In this lesson today, we are going to discuss transport across cell surface membranes. We'll discuss its outline first, or an introduction, to transport of materials across a cell surface membrane. This is, for instance, an animal cell. It's an animal cell. You have already understood in your previous class that there are many substances that move into the cell and there are substances that move out of the cell. Essential substances that move into the cell that include maybe oxygen, glucose, amino acids, fatty acids and glycerols, you already know there are ions that move into the cells, like sodium ions, maybe chloride ions, potassium ions. These substances also, or these ions also move out of the cells. Sodium ions can move out of the cells. Chloride ions also can move out of the cells. There are some waste substances that that move out of the cells that include maybe carbon dioxide. It may be some hormones, and maybe enzymes that move out of the cells. And above all, water moves into the cells and water moves out of the cells. Okay? So there are some essential substances that move into the cells, and there are some waste substances or some cell secretions that move out of the cells. We will discuss the methods that are involved in the transport of these materials into the cell or out of the cells. So let's see transport across cell surface membranes can be divided into two methods. You can divide it into two methods. It is passive transport and an active transport. Passive transport involves three methods. It is diffusion, facilitated diffusion, and osmosis. Whereas active transport can be divided into two types. One is through protein pumps, and other is through vesicles. Okay. In its strict sense, this active process, or instead of active transport here, I'll write active process. So there are two ways the materials are moved into or out of the cells by active process. One is an active transport, and the other is bulk transport. Okay. The very basic difference in between the passive transport and active process is that passive transport does not involve the use of energy, whereas an active process that involves the use of energy to move the substances into or out of the cells. So we'll discuss these processes one by one. So let's see diffusion first. To understand diffusion, we need to understand that gases, liquids, and solids, they consist of atoms or molecules that may be interacting with each other. The gases molecules have weaker interactions in between them, and they are randomly moving due to the energy which is known as kinetic energy. Okay, so if they have a higher rate of random motions, they have higher kinetic energy, and if they have lower rate of random motions, they would have lower kinetic energy. So these molecules are in random motion. Molecules or ions are in random motion due to their kinetic energy. So if these molecules are separated in two regions, like if this is region A and we have this region B, this is region A and this is region B. They have been separated into two regions. They may or may not have a partially permeable membrane present in between them. So in both of these regions, the molecules are moving randomly. So they would be moving in both directions from B to A. Molecules will be moving from B to A, and they will also be moving from A to B. So they will be mixing up together. For example, if there is high concentration of oxygen or any other gas, so this has high concentration here, and this has low concentration here. The molecules will be moving in both directions, but the net movement of these molecules will be higher from A to B, and they will continue to move until the concentration in both of these regions that becomes same. So these molecules will move from this region to this region, and the concentration will continue to increase on B side, and concentration that continues to decrease on A side. So consequently, what will happen? The concentration on both of the sides, that becomes the same. So an equilibrium is established. So this random motion of the molecules that continues until the concentration becomes the same on both sides. And this is known as equilibrium. This process of random mixing of the molecules from one region where they have high concentration to the region where they have low concentration without the use of energy is known as diffusion. Let's see an example. And before discussing an example, let's see how do we define diffusion. Diffusion can be defined as definition of diffusion. Diffusion can be defined as net movement of molecules or ions of a substance from a region of their higher concentration to a region of or ions of a substance from a region of their higher concentration to a region of their lower concentration due to their kinetic energy until an equilibrium is achieved. So let's see an example now. For example, we take a test tube. A large test tube, and we fill water into it up to this level. This is water filled into this level, and we put a whisking tubing into it. A whisking tubing is a tube made from a very thin membrane which is made up of cellulose membrane. It has very fine pores of molecular size in them, and are partially permeable. So we put a solution of starch and glucose in it. So this has a solution of starch plus glucose in it. So after we put this whisking tubing in this test tube, which already contains water in it, after 30 minutes or after maybe some time, after 30 minutes, we take water from outside the whisking tubing. Take water from outside the whisking tubing. And test for starch and glucose. And glucose. So when you test it for starch with an iodine solution, you will see starch test is negative. Starch is not found in outside solution, whereas glucose test is positive. It means that glucose has moved out of it. Okay, so this is partially permeable membrane. It allows some substances to pass out of it or move out of it, and does not allow the other substances to move out of it. How we can explain this mechanism? In this non-living system, the molecules of glucose and the starch, they are in continuous random motion inside. They have high concentration. This is high concentration inside, whereas there is low concentration on outside and low concentration outside. Starch molecules do not move out as the membrane is partially permeable. Starch molecules are larger molecules and they cannot pass through or move through the pores in this cellulose membrane or this whisking tubing. However, glucose molecules are smaller and they can move through the spaces or the pores in this partially permeable membrane or in this whisking tubing, down the concentration gradient. So that is why after 30 minutes, when we tested water from outside for starch, there was no starch found outside the whisking tubing, as starch has not moved out of it, whereas the glucose was tested positive on outside of it, as glucose has moved out of it, down the concentration gradient. We can extend this experiment over a period of time, and we can, test both the solution from inside and from outside the whisking tubing after every maybe 5 minutes for maybe 20 minutes. So after, if we take a sample from, this is sample from inside the whisking tubing and sample from outside the whisking tubing. Okay. At start of this experiment, at time You see, if I place time in minutes here, so at 0 minutes, you will see the sample from inside. Starch test and Benedict test. And similarly, starch test and Benedict test. So what will be your observations at 0 time? You will see blue-black color inside the whisking tubing for starch, and you may see brick red color when the solution from the inside has been tested. And outside the whisking tubing, this would have maybe orange color, and this would have blue color at the start of an experiment. After 5 minutes, however, you may find that the starch is still blue-black inside, but the Benedict test inside that gave us an orange color, and the starch test outside is an orange, and the Benedict's test that gave us maybe green color. After 10 minutes, this was blue-black, and this may be yellow, this is orange, and this is yellow again. Okay. So from these observations, we can conclude that the starch has not moved out of the whisking tubing. So the starch shows the blue-black color inside, and it shows the orange color on outside. So it means that the starch has not moved out of it. Okay. Whereas do you see the color inside the whisking tubing for the Benedict with the Benedict test shows that the glucose concentration inside is decreasing. So from brick red it turns orange, and then it turns yellow. And here do you see the concentration is increasing. Okay, and after 10 minutes it becomes yellow on inside and it becomes yellow on outside. So it means the glucose concentration between inside and outside, that becomes the same. So this is where an equilibrium has been established at this point. Okay, so this yellow color on inside and outside, that shows an equilibrium is established at this, or equilibrium is achieved after 10 minutes. Let's continue with the role of cell membrane in a diffusion. The role of a cell surface membrane in diffusion. As we have understood the structure of a cell membrane, we know according to a fluid mosaic model, a cell membrane has phospholipids arranged in a phospholipid bilayer. Okay. So if I draw the phospholipids, they have the polar heads and non-polar tails. Tails are facing each other and the polar heads are facing outwards, and this makes a phospholipid bilayer. So this is a phospholipid bilayer. Okay. The tails are non-polar. These are non-polar tails provide hydrophobic barrier. Okay. And polar heads are outwards. These are polar heads. So any non-polar molecules like glycerol can move from high concentration to low concentration. If it is high concentration on outside, it can move into the cell. Ethanol or an alcohol which is produced during anaerobic respiration in some cells, that can move out of it from high concentration to low concentration. Even steroid hormones, okay, that are synthesized within the cells, that can also move out of the cells. So if this is a steroid hormone, it can also move out of the cell down the concentration gradient from high concentration to low concentration. So if this is high in concentration here and this is low in concentration here, so it can move out of it through the spaces in between the phospholipid molecules. Similarly, an ethanol molecule can also move out of it from high concentration to low concentration. Okay. However, larger polar substances or water-soluble substances like glucose cannot pass through it. Glucose molecule, even if it is high concentration on outside, this is polar and this cannot move through the spaces between the phospholipids through a phospholipid bilayer. Even sodium ions cannot move through this from high concentration to low concentration, so they do not move through the phospholipid bilayer. We can explain why these substances do not move through the phospholipid bilayer. The non-polar tails provide hydrophobic barrier to the large polar molecules or ions to move through the phospholipid bilayer. Okay. Gases can move through. So polar smaller molecules or ions can move through phospholipid bilayer by diffusion. Non-polar molecules can move through the phospholipid bilayer by diffusion. Gases are an example. Gases move into or out of the cells by diffusion. We'll continue with the concept of concentration gradient. Concentration gradient. And then we'll discuss the factors affecting the rate of diffusion. We'll discuss how we calculate the rate of diffusion, and then the factors that affect the rate of diffusion will be discussed. If we divide a room into two different regions, like region A and we have region B, we can divide the regions like inside a cell and outside a cell. If a substance has high concentration in region A, if this is high concentration of molecules or ions of a substance on A side, and it is low concentration of the same molecules or ions on B side. So we can plot this concentration by a bar chart. On region A, it has high concentration. So this shows a taller bar. And on region B, it has lower concentration. So we can draw a shorter bar. Okay. On y-axis, we can label, this is y-axis, and we can label concentration on this y-axis. Okay. So there is a difference in the concentration on both sides. This can be calculated by plotting a slope from high to low concentration. Okay, this slope or the concentration difference in the concentration at two regions, this is known as concentration gradient. So concentration gradient can be defined as concentration gradient can be defined as the difference in the difference in the concentration of molecules or ions of a substance between two regions. Okay. The slope that represents the difference in concentration is known as concentration gradient. You can see a relationship in between the difference in the concentration and the steepness of the slope. The steepness of the slope, is proportional to the difference in the concentrations. Greater the difference, steeper the slope. Okay. So steepness is proportional to the difference in concentration at two regions. In the next part, let's see how do we calculate the rate of diffusion. The rate of diffusion. Rate of diffusion of molecules is very slow, and that's why it is possible on very short distances only. Over long distances, the diffusion becomes very slow. That is why the cells have a very smaller size, so that the diffusion can take place in them. If the volume of or the size of the cells had been larger, the cells might not have been able to survive due to the slower rate of diffusion. So rate of diffusion determines actually the size of the animals or the plant cells or the other cells. How do we calculate the rate of diffusion? The rate of diffusion can be calculated as the change, maybe the distance moved by the molecules by the time taken. Okay. Let's see the factors the rate of diffusion. There are many factors that affect the rate of diffusion. They can increase or they can decrease the rate of diffusion. That include number one, steepness of the concentration gradient. Number two, size of the molecules. Number three, the distance over the diffusion has to take place, or the thickness of the exchange surfaces. And surface area of the exchange surfaces. So let's see the effect of steepness of the concentration gradient on the rate of diffusion. If we have a higher concentration of a substance on region A and lower concentration on region B, though molecules will be moving in both directions, but the movement of molecules from direction A to B will be higher. So the net movement will be from A to B. The rate of movement of these molecules depends on the difference in between the concentration on these two regions. If there is higher concentration on this A side and it is lower concentration on B side, so this would have, this a concentration gradient. This is a concentration gradient. So molecules will move from A to B direction down the concentration gradient without the use of energy. If we make a difference in the concentrations on both sides in such a way that the concentration on A increases and B decreases, this would affect the concentration gradient. Okay? So if this is, concentration increased further on region A and concentration decreased on region B. Okay? So what will happen to this concentration gradient? This will become steeper. Okay? So this is a steeper concentration gradient. If we observe the changes, or we can experiment to see the movement of molecules from a region of higher concentration to the lower concentration, we will find a higher rate of movement of molecules if the steepness of the gradient is increased. So we can say steeper the concentration gradient, higher the rate of diffusion, and vice versa. Then the second factor of the size of the molecules or the nature of the molecules, effect of nature of molecules on the rate of diffusion. Larger molecules have slower diffusion rate, of diffusion. Why? The reason is that larger molecules need more energy to increase their random motion. So they need higher kinetic energy. So that would slow down their movements and reduce the rate of diffusion. Similarly, non-polar molecules can easily diffuse through the phospholipid barrier than polar molecules. Surface area also affects the rate of diffusion. Greater the surface area to volume ratio, higher the rate of diffusion. Greater the surface area to volume ratio, higher the rate of diffusion. And finally, effect of temperature. Effect of temperature. Temperature increases kinetic energy of the molecules and increases the rate of random motion. So, temperature increases the rate of diffusion. I'll write increase of temperature increases kinetic energy. Higher the kinetic energy, higher the rate of random motions, so higher the rate of diffusion. So that's it for now. For practical demonstration,